All right, thank you everybody who came to the InterSTEM Research Symposium. Just some team introductions. I am Simone. I'm one of the program directors of the InterSTEM Research Program. Also here is Arul, who also runs the program. And over the past few months, we have been leading the biology and math team as they have developed their research papers in their own respective topics. So the first team was biology, and they performed genetic editing for breast cancer research. And I will turn it over to Arunima to share your slides and tell us a bit about your research.
Okay, so basically our group chose to focus on how biotechnology and CRISPR can serve as a potential treatment for breast cancer treatment. So the members are Chang, Sohum, Mehta, Krrisha, Patel, and Sophia, Zheng.
All right, first off and foremost, hello everybody. Very glad to be here, and I will be giving a brief introduction to the context of breast cancer. So next slide, please. All right, so as the name suggests, breast cancer is basically cancer that affects breast tissue and the surrounding tissue. And it is the second highest cause of mortality in women of all races and ethnicities. So for us, we decided to do the research on CRISPR, which basically stands for clustered regularly interspaced short palindromic repeats. And these can be used to genetically edit the cancer cells. So when DNA is added, replaced, or edited inside the current genome, this is referred to as genetic editing. And CRISPR has the ability to eradicate tumor tissue by deleting the lipocalin-2 gene, which is linked to breast cancer. And CRISPR can enable the acquisition or deletion of mutations, allowing researchers to pinpoint the location of the causative gene of whatever is causing the breast cancer. So this can definitely aid in the identification of whatever problem genes there are, like what's causing the cancer. And it can also lead to the development of further gene therapy further down the line. Next slide, please. All right, so next slide once again. But yeah, this is going to be our basic method. So yeah, so obviously the most well-known one that you guys may think of is just direct implementation. But one of our sources heavily indicated that in vitro was also a method that was used. So essentially, when researchers are growing cell cultures, they can obviously grow breast cell tissue, but then they would use that CRISPR system to deactivate tumor suppressors. And for the examples that we had over here, it was APC, TP53, and SMAD4. So when you deactivate these tumor suppressors, you have that kind of opportunity to discover how the progression works within the cell culture. So not only is this more accessible, it also helps out with discovering newer methods of cancer treatment, whether that's newer methods of therapy or anything else. So this helps mitigate ethical concerns, as I'm sure you guys are all aware of the various debates about monkey and mice being used in drug trials and whatnot. And compared to human trials, which is like requires a lot of necessary paperwork, this is more relatively accessible. Yes, sir. So yeah, so this is probably the implementation of CRISPR that is the most common, and it's also what you guys would probably think of. So yeah, so genes in a cancer cell could be altered to undergo apoptosis, and obviously the reasoning behind this is because if you obviously have a cancer cell, you would want to eliminate it as quickly as possible. So various genes could be altered, and this would undergo premature apoptosis, which is definitely the desired result, as you do want to limit the amount of breast cancer tissue is in there. So there's more. So the methods, because it is lower efficiency, the more recently proposed methods are electroporation, microinjection, and adeno-associated viruses. And it's a far more direct process than in vitro, where you're just looking at the progression, and it is, I guess you could say that it is directly happening at the same time. And I'm going to give a brief rundown about what those methods actually mean. So electroporation is poration, as you can see within the image on the right, and essentially the use of electrical pulse to increase the permeability of a cell membrane. And this way you can just directly insert that CRISPR, plasmid gene inside of there. And microinjection, as the name suggests, it is just basically you're injecting, as I said, a relatively small plasmid gene into the desired cell. And the adeno-associated viruses are small viruses that can be used for gene therapy. And the reason that it is strongly suggested by researchers is due to its relative stability.
Okay, so thank you, Gordon, for describing all the methods. So now I'm going to be talking about the results that we found and how we analyzed the evidence to come to our final conclusion. Okay, so we finalized that CRISPR effectively, one, resists cell death, evades the growth, and also performs artificial apoptosis successfully. So here we have a picture of how the CRISPR and the genetic editing is done. So here we have the RNA target device and then the Cas9 cutting device. So here that's when they genetically modify the cell and provide a cell therapy, and the RNA recognizes and binds to the target DNA. And then in the last step, the new genetic material is finally produced, which is going to be used for future attacks. So basically, when applying this method, there's a strong memory retention of the application, which is why it's so effective. So CRISPR allows the bacteria to store the previous viral and plasmid attacks to use as self-defense for the future attacks, and that's why it's so effective, because it has such strong memory retention. And then during an in vivo trial done by Dr. Sandmer from the National Cancer Institute, the treatment was evidently working for a patient. So for example, the tumor had stopped growing because they were able to genetically edit the cell to ensure it knows how to prevent the growth during future attacks. And hence we came to the conclusion that the CRISPR techniques can be used to treat solid tumors through cell therapy by reducing its growth substantially.
Based on the results, it has allowed us to formulate a summary of basically how CRISPR can be used in the future years, and it allowed us to also provide some limitations. So this study does have some limitations which need to be mentioned and interpreted carefully. First and foremost, genome editing is a relatively new concept that was first found in bacteria and archaea, which are defined as prokaryotic organisms. This application of four systems into the human body is a relatively new advancement, but it also led to some restrictions. According to the National Library of Medicine, a relatively high frequency of off-target effects, otherwise known as OTEs, have been observed at a frequency greater than 50%. This occurs due to the natural tendency of Cas9 unintentionally binding to unwanted genomic sites for cleavage, and this can lead to consequential mutations. However, multiple methods and strategies have been formulated in order to reduce the frequency of OTEs, such as improving the single-strand RNA specificity and optimizing guide designs. Furthermore, another limitation is constraining the delivery. Conventional gene therapies that utilize viral vectors are not 100% efficient, and this can lead to immune reactions and insertional mutagenesis. And this is basically when DNA sequence from outside of an organism integrates with the genome of a host organism, and this can lead to the creation of mutations. And lastly, another limitation is DNA damage toxicity. Double-strand breaks that are induced by CRISPR have resulted in apoptosis, otherwise known as programmed cell death, which is obviously not the desired result. The desired result is a gene edit within target sequence. However, future implications of CRISPR-Cas9 indicate a very positive future. For example, CRISPR-Cas9 has allowed for precise and direct edits to the target DNA sequences, and this was not available at a very low cost and very efficient rate prior to finding CRISPR-Cas9. Also, multiple genes can be corrected, removed, replaced, or inserted in vivo in continuous manners. And it can also serve as a therapeutic option to target the immune system against cancer, which was the primary topic of this research project. And lastly, CRISPR-Cas9 provides a tool to manipulate non-coding regions of the genome, and it can accelerate the functional exploration of the cancer genome.
So our conclusion is that after looking through all the results and the potential benefits that genetic editing can bring, we believe that there are a few main advantages of trying to use CRISPR editing for specifically genes in breast cancer. It can lead to a faster and a more accurate diagnosis. It can help with targeted treatments and that are more specific towards the patient, so more individualized treatments, and it may also result in a lower cost in the long run for patients. As we stated again, the downsides of genetic editing are that there still needs to be more research done on the exact benefits that are implicated, and that there may be some ethical concerns and testing regarding this.
So for our next research team, it was a math team, and they also looked at breast cancer, but instead of CRISPR, they looked at immunotherapy and its effect on breast cancer growth using mathematical models. And then now I will turn it over to Cayden to present his team's research findings.
Hello, everyone. Our topic is analyzing immunotherapy and its effects on breast cancer growth by myself, Cayden, and Daniel. To begin, we have the abstract. And so basically, the immune system is a vast system of cells, organs, and tissues that serve as a primary defense mechanism against health threats, mainly, well, for the purposes of our research, breast cancer. And as we know, white blood cells, also known as leukocytes, are also integral to our body's defense mechanism, fending off various diseases that are harmful to our bodies. Coming from our bone marrow, these white blood cells travel throughout our lymphatic system, which serve as our body's sort of like sewage system, and also maintains the fluidity of our body's tissues through through removing toxins and fluids that leak from our blood vessels. It is through these mechanisms that cancer is fought, and immunotherapies work hand in hand via these systems. And immunotherapy, white blood cells detect antigens on the surface of cells, and they distinguish between healthy cells, foreign invader cells, which we'll also get into later on, and cancer cells, as these receptors are all different in both. These immune cells will secrete antibodies to program these antigens for destruction, killing off these harmful cells. And some cancer cells, however, can avoid this detection by producing fewer antigens, producing checkpoint proteins, or by slowing down or stopping the immune system response altogether. Immunotherapy, working against these cancerous and harmful cells, utilizes the production of antibodies made in labs. In this paper, we examine, in this presentation and the paper that we wrote, we examined the effectiveness of immunotherapy drugs against triple-negative breast cancer, which is the one sort of branch of breast cancer that we're primarily focusing on here, and the plausibility of its use as a replacement for traditional treatments, such as the more popular therapy called chemotherapy. We found that a leading factor to consider in predicting the effectiveness of immunotherapies is the number of prior therapies a patient has undergone, which we'll also get into later. The expression of PD-L1, which is a protein expressed by tumors to avoid immune detection, plays a major role in immunotherapy potency, as higher expressions were shown to correlate with higher effectiveness. Immunotherapies also work well in conjunction with chemotherapies, which will be part of our results later on, especially if the patients express the PD-L1 protein.
All right, so I'll be talking about the introduction. So cancer is a disease where mutated cells that undergo unregulated cell growth have the potential to spread and invade to other parts of the body. Cancer spreads through a process called metastasis, where some cells split off the primary tumor. Oh, yeah.
Oh, sorry, I forgot to mute.
Okay. Escaping the tumor provides mobile cells an environment where without competition over nutrients and space, and cancer cells spread through the epithelial tissue to the other tissue to other organs, and epithelial markers are downregulated while stem cell and embryogenesis factors are overinduced, which increases self-renewal capabilities. And when a cell is defective, apoptosis is triggered, and in cancer cells, mutations in genes that trigger apoptosis prevent these genes from properly signaling, which results in 2 nuclei per cell, distortion, and abnormal shapes. So breast cancer, which is the focus of our paper, often begins developing in milk production ducts, which is called invasal ductal carcinoma. And it is also common for breast cancer to begin in glandular tissue called lobules, which is named invasive lobular carcinoma, and in breast cancer, cancer cells commonly metastasize to nearby lymph nodes. We will conduct our research by analyzing and comparing online data pertaining to our focus, and then using this data to develop our conclusions. Through our research, we aim to further explore immune response to the development of cancer cells in the breast, dig deeper into the body's attempt at regulation and suppression by means of the immune system, and we will also explore the various protein pathways and interactions that are closely involved in the process of cancer response. So on to our methods. Cancer and its methods of prevention have long been topics of varied and extensive research. In particular, because of the higher level of difficulty in detecting breast cancer, we chose to focus on breast cancer to develop more knowledge regarding the effectiveness of therapies. We began by comparing data of different types of breast cancer, including ER+, HER2+, and triple negative breast cancer. From our findings, specifically in regards to triple negative breast cancer, led us to mainly select patient data from triple negative patients. And we also obtained and analyzed data on the usage of drug immunotherapies against triple negative breast cancer, allowing us to identify common trends and themes. Despite minor varying and unexpected findings, we found success in drawing conclusions on their corresponding effectivenesses. In order to truly assess the benefit and effectiveness of immunotherapy, we also compared and utilized papers comparing immunotherapies to other techniques and forms of cancer treatment, most notably chemotherapy. Part of our approach towards the comparison included considering the complications and downsides of each of the treatments, in addition to just the pure difference in effectiveness. However, we did not only juxtapose these different treatments against each other, but we also examined the possibility of a combination of treatments and how various therapies would coexist and work together.
Okay, so onto the results. Sorry. So onto the results, right? As we know, immunotherapy goes after the resistance mechanisms that breast cancer typically develops through when they undergo cancerous growth and spread throughout an individual's body. By using the patient's immune system to target their breast cancer and unwanted foreign invaders, immunotherapy works to alleviate and halt further harmful growth. After the collection of data via the methods discussed by Cayden earlier, we've concluded that these immunotherapies have far less side effects when compared to other forms of cancer treatment, namely chemotherapy again. This is strictly due to the fact that immunotherapy solely targets an individual's immune system rather than the individual's, like all of the individual cells. And so immunotherapy largely does its job by detection of foreign mutations encoded within these cancer cells, whereas other types of cancers like lung cancer and melanoma have many noticeable mutations in their DNA. Uh, in breast cancer, mutations are less noticeable and thus is harder for the immune system to recognize. So moving on, as we know, breast cancer is typically classified into three categories. As we can see here, ER plus, HER2 plus, and triple negative. And as we mentioned earlier, our focus is primarily in the triple negative section of breast cancer. And we call that TNBC for short, triple negative breast cancer. And we're using this branch of breast cancer in particular because through much experimentation and results and from credible sources that we found that triple negative breast cancer typically has more noticeable mutations in its DNA. So it's easier to detect and easier to trace in our results and in our graphs. And so there we have that. And this graph also models that as well. The far right is the triple negative breast cancer, also known as basal-like breast cancer. Those two are very similar. And so as you can see here, the variations are clear. These on the far right, we have more mutations than those on the left and a larger margin of error as well. and it has less outliers and extends higher up. And so, yeah. Also, we found that um, results also reveal that TNBC also has a higher rate of expression of PDL1, which is a receptor on the tumors that is expressed to evade immune detection. However, however, antibodies were made in response to these cancer cells. They worked to an extent after experimentation, but not as well as they initially expected, because of effects varied amongst patients rather than being stable across the board. It was also found that these agents worked better with patients that hadn't already had a lot of prior therapies, as shown in this graph. This is sort of a hypothetical graph that shows the effectiveness of these immune agents and drugs um, in response to patients with a various number of therapies that they've had in the past. And so, as you can see, if a patient has never been treated with immunotherapy before, the agent effectiveness would be higher, and the opposite holds true as the number of therapies increases. And so on to Cayden.
All right, so as we can I'm not sure if you can see that, but as we can see in that visual, it's not showing that, but okay. There's like about 20% increase in effectiveness if, like Cayden said, if the immunotherapy was like their first treatment. And we'll next we'll be comparing chemotherapy to immunotherapy treatment effectiveness, and a comparison on TNBC yielded that there's a slight difference between the two, and although the difference may seem insignificant because the curves are similar, patients with a higher expression of PDL1 were actually better off and found better results with immunotherapy. Some side effects that we saw were organ toxicity, and that the immune system can become hyper energetic and can cause misdetection of foreign mutations, which can lead immune cells to flood healthy organs, causing inflammation. Okay, there was supposed to be a visual too, but which can cause inflammation, which is caused by immune cells attacking healthy parts of the body. Another trial was also done where chemotherapy and immunotherapy where they tried to use both at the same time, or not at the same time, but like on the same patient, and they found that the two treatments work together hand in hand, because chemotherapy killed some of the cancer cells and released foreign proteins from these cells, which helped immune system recognition, which then allowed immunotherapy to work even better and function more easily. And the results also revealed that patients with PDL1 receptors on cancer cells had a better time with both drugs as opposed to only one treatment or only chemotherapy, because those who use these methods typically lead longer lives.
Okay, so yeah, that's basically all we have for now. We're just missing the conclusion and the discussion part, as we mentioned earlier. Also, were my slides, like, were you guys able to see my slides moving or no?
Oh
No,
yeah.
I don't think so. Like the graphics were glitching a bit.
Oh, really? Okay. I'm sorry about that. Yeah, because I was like flipping through them and I thought we could like trace them, but okay.
No,
Sorry
it's
about
okay. It's okay.
um
If you could stop.
screen sharing. Okay.
I think I got it. Okay, thank you all so much. We're just about to be done here. Thank you everybody for participating in the research program, and congratulations on finally finishing your research paper and presenting it all.